Hello, so I've been asked to redo a video on the neuromuscular junction, neuromuscular transmission, how a neuron talks to a muscle. And really before we do this, I got to kind of go back and just recap a couple little things and remind ourselves about a simple topic here, not so simple really, is a synapse. And really what is a synapse? That's when two cells come together for communication with each other. So there's going to be some kind of communication occurring between two particular cells. In this case, this particular synapse is called a neuromuscular junction, or NMJ. So there must be a neuron and there must be a muscle fiber, right, or a muscle cell. So there is two cells, neuron and muscle, who are going to talk to each other at a synapse. So what we got to do is we're going to begin really looking at the structure of the synapse, getting an idea what's going on, then look at its physiology, as this will be a pretty lengthy video compared to most. So remember, we have a neuromuscular junction, we have the synapse between a neuron, and then there is a muscle fiber, and this being a muscle fiber, right? Muscle cell, muscle fiber. And these two cells must communicate with each other. Now, to do so, we want to understand that this is a synapse called a neuromuscular junction. It takes two cells to have a synapse. So we have a neuron and we have a muscle fiber. Let's think of the neuron, because in this, the neuron comes first. This is pre-synaptic, which means it comes first in the synapse. Muscle fiber, because it comes second, is the post-synaptic cell, as it comes second in the synapse. And there is a gap between these. Now this gap always exists in special synapses called chemical synapses, called a synaptic cleft. Now you might remember in lecture we talked about this in terms of a relationship, and we said, okay, now we want to imagine that the relationship, this case, the neuron, is the wife, right? She comes first and she tells the husband, the muscle cell, what to do. Remember, hubby doesn't know how to do anything unless the wife tells her to, right? The wife has to be able to tell the husband what to do. So this neuron, being presynaptic, is controlling the postsynaptic cell, the muscle cell. But if they were too far apart, diffusion would not be efficient enough to take neurotransmitters to the receptor sites on the muscle fiber for it to work. If it was too close, diffusion, it just wouldn't work. It has to be just right, and that just right space or gap is called the synaptic cleft. Now, because the wife, the neuron, is in charge, she has her honey-do list. She has a variety of instructions for the husband, the muscle fiber, to do. In this case, they are contained in vesicles, and inside these vesicles is a particular neurotransmitter called acetylcholine. I'm going to abbreviate it ACH for short, acetylcholine. And acetylcholine, ACH, is the particular neurotransmitter found in the neuromuscular junction. Now, as well in the neuromuscular junction on the muscle fiber, you're going to have a very specialized area here. And this very specialized area is called a motor end plate. And the motor end plate is unique as it possesses receptor proteins that are needed for communication with acetylcholine. And these receptor proteins are called nicotinic, as in they bind to nicotine, the molecule, the drug, the chemical that is found in cigarettes, in tobacco, nicotinic, acetylcholine receptors. So these nicotinic acetylcholine receptors are found on the motor end plate, and given this, to give more of a surface area, there are folds located on this called junctional folds, and the junctional folds have the job of increasing surface area for nicotinic acetylcholine receptors so they can interact with the acetylcholine. Now, Given this, just a couple reminders. Remember this side of the neuron right here that we're dealing with is part of the neuron here. It's called a synaptic terminal. The cell membrane of a muscle fiber is called a sarcolemma, and those are terminologies we talked about previously. So what we want to do is kind of come back to this drawing of our NMJ, and we're going to use our neuromuscular junction. Let me give just a little bit more space here for drawing. And we want to make sure we got our junctional folds, our neuron, our muscle fiber, and we want to start thinking about what's going on here. So remember, we have a motor end plate with nicotinic acetylcholine receptors located there. We do have vesicles, and these vesicles are containing a neurotransmitter called acetylcholine, and that is bound up and stored in these vesicles. Acetylcholine. What we're going to see happen is, first off, our muscle fiber, remember, there is a fluid inside the ICF, which we also call the sarcoplasm in muscle cells. Muscle fibers are sarcoplasm is a special name for the fluid inside of a muscle cell, as if in the muscle tissues, muscle cells, we are dealing with unique physiology, so we want to say sarcoplasm instead of cytoplasm. And the fluid outside, extracellular fluid, or ECF, and we got these two fluids. Now remember, like all cells, muscle fibers have a protein, the sodium-potassium ATPase pump. And the sodium-potassium ATPase pump, as its name implies, pumps sodium and potassium. The sodium-potassium ATPase pump is going to bring in 2 potassium cations and pump out of the cell 3 sodium cations, Na+. And what you're going to notice is the ECF maintains a very high sodium concentration but low potassium, where the ICF has a very high potassium but very low sodium concentration because of the sodium-potassium ATPase. Given that, the ECF and the ICF will have a unique difference of charge. Why is that? Well, the ECF has more cations outside than cations are inside the ICF. So there is a greater positivity outside in the ECF than in the ICF. The ICF is actually slightly negative. Just for posterity, it's about negative 95 millivolts in charge at this point. And the point that I want to make, not so much the charge, but the fact that we refer to this cell as being polarized, as it is negative in, positive out. Right? Remember, when you are polarized, you are negative in, positive out. Right? So what we got with this cell is, we would say he is resting. The muscle is not contracting, not doing anything. What we want to do is make this muscle do something, make it contract, and remember, to do that, we must have a neuron to tell it to do that. So we've got our receptors, or nicotinic acetylcholine receptors. We've got our vesicles, and our vesicles store the neurotransmitter acetylcholine. Acetylcholine, I'm going to abbreviate it here, ACH again, acetylcholine. And we've got our nicotinic acetylcholine receptors, NACH receptors, right? Very hard to write. Receptor, they're that small. So remember, this muscle cell is polarized, right? Because the ICF had a very high concentration of potassium but low concentration of sodium, made this negative. The ECF, who has a high sodium and low potassium, is more positive, and it's polarized. Well, we're going to stop that. We're going to make that not be the case anymore. And what we want to do is, starting off in your brain, in the in your frontal cortex of your frontal lobe, prefrontal cortex, you produce a signal, a neural impulse that travels down the spinal cord, out the spinal cord, through a particular nerve, out to a particular set of axons to a muscle to tell that muscle to contract. And so this electrical impulse travels down the neuron. And as it travels down the neuron, this electrical impulse is called an action potential, AP for short. And this action potential, as it arrives, is a sudden and very drastic change in voltage. And there's a very special kind of protein here that we would have embedded in the synaptic terminal. And these proteins are what we say is voltage gated. What do we mean by that? It is only opened by a particular voltage, and they are voltage gated Ca2+, or to say Ca++ calcium cation. So they're voltage gated calcium channels. Now, why is it plus plus? Has 2 extra protons. Remember, calcium only has 2 valence electrons. It gives up those 2 valence electrons, become a cation, and the cation having a positive charge because of 2 extra protons here. So calcium diffuses in to the neuron, into the synaptic end. The calcium cation, Ca2+, diffuses in, and it will allow and cause for the exocytosis of the vesicles, acetylcholine. It will cause exocytosis. Now, the exact mechanism in this course we are not responsible for, but you may be responsible for that in later on courses. But acetylcholine dumps out into the synaptic cleft, and acetylcholine binds to the nicotinic acetylcholine receptors. Now, what I want you to pay close attention to is this, and why this is happening is that this will ultimately open up a very important kind of gate. And these are chemically gated sodium channels. Now, if you remember from my lecture, I showed you guys that these are part and parcel of the same protein, that basically there's a receptor surface and there's a conducting surface of the ions. So what we're going to see is this voltage, or the chemically gated sodium channels, they open. Now, we got to figure out which way is sodium going to go. It could go in, it could go out, and the trick is to figure out where it's going to go. Now, remember, these are channel proteins that let ions go through them. Ions, being not lipid soluble, must go through a protein. Remember, a protein is needed for facilitated diffusion because of anything that will not go through a cell membrane. So the trick is that we go from high to low through a protein. So the sodium will go through the protein because it has been produced at a high concentration gradient, and sodium comes in, right? Sodium comes in. Now, why that is? High to low, right? Facilitated diffusion. There's another reason. Sodium is a cation, Na+. If we look at that, Na+ cation, there's a positive charge. The ICF, remember, is actually slightly negative, negative 95 millivolts, right? It's quite negative right now. And if we notice it's quite negative, opposites attract, don't they? So what's going to happen here is there will be a sudden change in charge where it goes from being negative to being more positive, and we say this cell is depolarized. It is no longer polarized, but it has become depolarized. And if you guys can remember my silly song that I sang to you guys in class, remember my sodium gates bring the sodium to the sound. They're like, I'm depolarized, you dang right. I'm depolarized. I can teach you. I have a positive charge. This cell, being more positive, becomes to be depolarized. Remember we said when a cell is depolarized, it gets excited. And when it gets excited, an action potential generates on the sarcolemma of this muscle fiber. And this is where things are going to get incredibly interesting. as we have extensions on a muscle fiber called T-tubules, right? And the T-tubules possess terminal cisternae. Remember, my favorite ion is in here, Ca2+, calcium cations, Ca++, very hard to draw that on here, calcium cations. And we're going to see this action potential arise to the T-tubules, and it will cause the release of calcium cation from terminal cisternae, right? And this will cause a muscle contraction. And a lot is going on here. A whole lot goes on during this process. And it does get a little bit more complicated than this in reality, but this is the big, ultimate beast of what's happening. So we want to make sure we understand that. Now, the thing that's very important for us to remember here is that depolarizations are when we get more positive, as it moves in a more positive direction. To kind of give you guys an idea of what happens is that you've got this voltage, right, that we have, where we've got 0, and then we got a very negative charge and a much more positive charge. Well, negative 95 is where this neuron would rest at, right? And the neuron's sitting here at negative 95, not doing anything, and then the sodium levels come in, and it shoots up really positively in a very positive direction. So basically, you're polarized when you're at this resting potential. You get depolarized as you go up, okay? Now, what I want to also show you is that this depolarization forms an action potential on the sarcolemma. And this action potential, you may remember that T-tubules are what brings the action potential down into the muscle cell, where the terminal cisternae, T-tubule, two terminal cisternae called a triad, and this is going to regulate the calcium release right onto the zone of overlap in the sarcomeres. Now, to fully comprehend all that's going on there, basically this will stay this way, basically, as long as calcium is dumping out and being released. Muscles can pretty much stay contracted until really death and the proteins degrade and everything. So we want to stop this. And so this graph, we want to make this graph go back to a more negative direction again, so we got to learn how we do that. Now, in lecture, I would have mentioned to you guys that we have an enzyme that resides in the synaptic cleft. And this enzyme is called acetylcholine esterase, ACAG abbreviated here. And acetylcholine esterase has got a very important job to do. What acetylcholine esterase does is it's an enzyme who degrades acetylcholine that is bound to these receptors. So here we'd have our acetylcholine bound to the receptors, and acetylcholine esterase will degrade it, break it down, and then what's going to happen is once we break that down, our chemically gated sodium channels cannot stay open anymore. So they shut, and the cell is still depolarized, but it can't get any more positive. So it is depolarized still. Okay? What we want to do is undo that. So acetylcholine esterase, as it degrades acetylcholine, the choline it breaks down goes back, if I can write that again, there is a molecule called choline. And if I could spell, that's always a problem there, right? Choline. And then the other thing is it produces acetate. The acetate diffuses away. The choline is brought back into the synaptic terminal to be turned back into choline recycled. Now, what we want to do, we degrade acetylcholine, and that's going to shut the sodium gates. And then also, another very important thing that begins to happen is you've got a set of gates called voltage gated. Oops, meant to use this color. Voltage gated potassium channels, voltage-gated potassium channels. The potassium leaves. So it's no longer depolarized, it becomes repolarized. So what is happening in this case is it's going back down in a more negative direction, polarizing again. And you'll see that far more in depth when we get into neurons, when we really try to understand neurons. So what's going to occur here when this happens is, remember my T-tubules, I quit using the wrong colors, my T-tubules and my terminal cisternae, the calcium cations will go back into the terminal cisternae, okay? And what will happen there is the muscle relaxes, okay? Now, a few things that I always like to do is have the students really think about that, but what I like to do is kind of go back through that just really kind of briefly and help